Okay, we're going to put it all together now and really see how PCR makes millions of copies of a DNA molecule. So we start off with our double-stranded DNA template. So this is the thing we want to copy. And now I have it as a pretty short thing drawn on our notes here, but remember this could be a very long piece of DNA that goes in millions of base pairs in both directions. Now, at the end of PCR, what we're going to end up generating is, we do PCR, and we're going to end up generating millions of copies of this smaller fragment that's derived from the template. So we're going to make millions of copies of this. And that's why we refer to this as not just as copying, but as amplification. I think when you think about DNA replication, then you're making two copies of the DNA molecule. But in PCR, you are amplifying a small region of the DNA. So just some more terminology, this little fragment that we amplify is sometimes referred to as the amplicon, or also sometimes just referred to as a fragment, a PCR fragment. Either are correct ways to refer to it. Now, of course, we see these sequences at the end, and those are important for defining what actually is going to get made. All right, so now let's think about how we set it up. Of course, we're going to have our template DNA inside of our reaction. And now we're also going to add these two single-stranded pieces of DNA shown down here. And these are important because these are going to act as primers for the DNA polymerase. All right, so DNA polymerase needs to have a primer, and these are short DNA molecules. Now with one of them, you can see pretty easily where it's going to be complementary to the template, right in this little area up here. But the other one is a little bit harder to see. The primer is written from the 5' to 3' direction, and that's pretty standard for primers. But to see where it binds, you actually have to flip it over and then draw it in the 3' to 5' direction. So with the 3' here and the 5' there, then the sequence would be AATG, right? We just flipped it around. And now you can see that it's going to be complementary to this region over here. Okay, now in addition to the primers, we're also going to add, of course, the DNA polymerase. We need to add some buffers, and these are just going to create the chemical conditions that help the polymerase be active. And then we're going to make DNA, so we need A, G, C, and T. We need nucleotides, and technically these are called deoxynucleotrophosphates, or DNTPs, and so those are going to be added in as well. Now this all goes into a little tube, and then that tube goes into a machine called a thermocycler, which is going to change the temperature of this mixture. Okay, so here's a graph that depicts what we're going to call one cycle of PCR. You can see we have temperature here on the y-axis and time on the x-axis to give you an idea of how long each step takes. All right, so we start off pretty hot. It's up here usually between 94 degrees and 92 degrees. And this is all in Celsius. So that's very close to the boiling point. And this is called the denaturing step. And what happens with denaturing is all those little hydrogen bonds that hold the DNA molecule together break down. So we're going to go from double-stranded DNA to single-stranded DNA. So now we have single-stranded template molecules in our mixture. Okay, next we cool everything down. And there's a bigger temperature range for this next part. It usually ranges from around 45 degrees Celsius all the way up to 65 degrees. And we'll talk later about how we decide what temperature you want to use. So this is called the annealing temperature. And annealing just refers to single-stranded DNA molecules coming back together to form double-stranded DNA. Now, that happens all the way across, but remember we have those primers in our mixture as well. And so those primers, this is the part where they are going to bind or anneal to the template molecule. So our primers are going to get stuck on there to set the stage for DNA polymerase. Okay. Now finally we have this last part called the extension step. And you can see I made this one intentionally kind of different because this one isn't as set as far as its time goes. The temperature is usually pretty set around 74 degrees. And this is the step where DNA synthesis is actually going to happen. Now 74 degrees is important because that is the optimal temperature for the DNA polymerase that we use when we're doing PCR. So this is the optimal temperature for the polymerase. Now the length of this, oh yeah, well let's think about the 74 degrees first of all. So that's pretty hot, right? 74 degrees. I remember up there in the denaturing step, we're at 94 degrees, very close to the boiling point. So we have to use a special DNA polymerase to do this, one that isn't going to get denatured at these high temperatures. So we need a polymerase that's very thermostable. And so the most common one is a Tac polymerase, and the name Tac comes from the abbreviation of Thermus aquaticus, which is the bacteria from which this is derived. Now, these were discovered in the hot springs at Yellowstone National Park. So here we have volcanically heated water that's very close to the boiling point, but these bright colors are bacteria living in there. And so their enzymes have to be very thermostable. All right, so, another thing to notice is I mentioned this is just one cycle of PCR, but in a PCR reaction, you're going to do this usually 30 to 40 cycles. And every time you do it, you're going to double the amount of DNA that you have. So after you get done with this extension step, you just loop back around and go back to the denaturing step and start again. You do that 30 or 40 times. The extension step is variable in length. DNA polymerase can do about 60 base pairs per second. So that's pretty fast, but the length of time of your extension step is going to be dependent on the size of the fragment you want. You have to make sure you give it time for that polymerase to go across the entire region. Okay, so now we're going to think about the individual cycles. So we've set up our PCR reaction with our template, our primers, our polymerase, and all the other reagents. Now we're going to imagine heating this up to 94 degrees, so we get denaturing, single-stranded templates. We cool this down, let's say to 55 degrees to anneal, and that's what's shown below. You can see that we have those primers annealing to the template. Now outside of this region, this DNA is going to come back and be double-stranded, but within this region, the primers are going to anneal onto the template wherever they are complementary. So now what I want you to do is pause the video and try and draw in what would happen if we heat this up to 74 degrees and you get the annealing or the extension step. So pause it now and try and draw in what's going to happen. Okay, well hopefully your drawing looks something like this, where we have these two longer molecules here that are part of the original template, but now we've created these two new strands that started with these primers and extended in the direction that my arrows show there. Okay, so that's the first cycle of PCR. Now, if we go back to our original drawing of what's going to happen overall in PCR, we start with the template molecule, and we're going to make lots of this molecules that are just defined by the binding of the primers. You can see we haven't done that yet, right? So that would be that little piece in there. But right now we don't really have that. And so we have to go on to the next cycle of PCR to start seeing how that's going to happen. All right, so once again, we denature everything at 94 degrees Celsius. So we have single-stranded templates. Now we're going to cool it down to 55 degrees Celsius, and the primers are going to anneal on there. Now what I want you to do, again, is pause the video, and starting from this image here, I want you to draw in all of the primers. For instance, there'd be one primer that would bind on in this spot right here, right? And so find all the other primer binding sites, draw those primers in, and then imagine what would happen if this were heated up again to 74 degrees Celsius and extension happened, and we added on to the 3' end of all those primers. Okay, so hopefully your drawing looks something like this, where we now have 4 double-stranded molecules in our mixture, and you can see where all of the primers were anchored for making these molecules. Okay, now even after the second round, we still don't have any pieces that are just our amplicon or our fragment that is flanked by those primer sequences. We don't have those yet even after the second cycle, so we have to go on to the third cycle. Now, if you can draw this all out if you want to, if you feel like it would be helpful, but in the third cycle, this is where we finally start making those little short amplicon fragments. So you can imagine the primer, for example, binding on here. Then when that gets extended out, we're going to make this second strand. We can fill in the sequence down here in this region. But now you can see this is the first time where we're actually getting our amplicon being produced. And there's another molecule in here that will also produce an amplicon when it gets its second strand synthesized. Okay, there is a nice video that shows this on Polylearn, a nice animation, and I would suggest that you watch this. It's going to walk you through multiple cycles of PCR, and you can see these amplicons starting to be created. Also on Polylearn is more background reading about DNA polymerases and some of their qualities that are important for researchers doing PCR.